This is TWIS, This Week in Science, episode number 865, recorded on Wednesday, March 2nd, 2022. How to pronounce science. I think I did it right. I'm Dr. Kiki, and tonight on the show, we will fill your head with bugs, personality, and words. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. Democracy, ability of a people to choose their leaders and hold influence over their government, is the greatest threat to world governments, at least to those governments that do not have elected leaders. While democracy is something we kind of think of as a given in the Western world, 37% of the planet's humans are living in authoritarian regimes. In these authoritarian regimes, the people have limited freedoms, highly controlled access to information, and essentially no rights that cannot be taken away from them. What we call misinformation is often the only information in these regimes. Try to imagine for a moment that if all of the information you had access to, if every media outlet, website, and social media platform only presented you with false narratives, then I could tell you there is no pandemic virus, no global warming, and no such thing as cancer. Not only did America win in every gold at every event in the Winter Olympics, but I, your glorious leader, personally competed in and won each of those events. While that can sound ridiculous, Ahmadinejad claimed Iran had no homosexuals. State media claimed Kim Jong-il didn't defecate. That North Korea had cured cancer and AIDS, and that they had found an actual unicorn lair. China posts around 500 million fake profile pro-government posts on social media every year. Right now, Russian media is reporting that they are fighting fascist Nazis, and that Ukrainians are meeting the Russian troops with flowers and homemade pies. And in the US, there was a recent attempt to overturn a democratic election with propaganda and fraudulent electors. 37% of your fellow humans are living under regimes of state-sponsored misinformation. 37% of your fellow humans cannot vote, cannot speak their minds in public, and do not have basic human right protections. 37% of your fellow humans will live and die without knowing the simple joys of This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about science. And yes, the show does go on even as there are many human rights offenses around the world. And it's good to acknowledge those and to know that we have the privilege of taking this time to appreciate science. On this week's show, we have a great show ahead, of course, and we are so glad that you're joining us today. I have stories about, what did I bring? I brought some science and policy. Yes, I did. I also brought a lot of bugs, and I brought some brains. Woo! Digging into them. Well, not digging into them, imaging them. It'll be fun. Justin, what'd you bring?
I have microbiome and energy levels.
All right.
I have just good news, cancer edition. And I have a bunch of fake news, two different fake news studies that use Twitter.
Fake news. Blair, what is in the animal corner?
I have masks and monkeys, or should I say, I guess technically masks and primates. So the animal science police don't come for me.
Be accurate.
There are monkeys in both stories, but my second story also has apes, so it would be more accurate to say masks and primates.
Interesting. One of my stories has got a overlap of apes and masks. Wow, but I don't think.
No.
The only apes in my stories are the human ape. So let's jump into the science right here, right now. You're here for this science program. I want to remind you that subscribing to the TWIS podcast, if you have not done so, is easy to do on your favorite podcast platform. Just go wherever you find podcasts and look for This Week in Science if you are interested in finding us as a podcast. We also stream weekly live on YouTube, Facebook, and Twitch. Again, look for This Week in Science. And we are also TWIS Science on Twitch, Twitter, and Instagram. Woo! All of these different ways to find us, but the best one is to go to our website, twis.org. All right, now all that bookkeeping is done. Whew. It's science time. Well, actually, it's science and policy time. I want to just dive right into the big elephant in the room. What is coming next for the U.S. and the world when it comes to science and policy? Just a day after the IPCC released on Monday its latest and most dire report on climate change impacts, adaptations, and vulnerabilities, really not a pretty picture, Biden gave a State of the Union address that spoke of climate change only twice with respect to economics, basically. And while the IPCC report warns of future disease spread, the CDC has released new mask guidelines that basically put the onus on the individual to be careful when it comes to wearing masks. And the White House just today released a new pandemic preparedness plan that actually does look comprehensive enough to help us through future COVID variants and maybe future pandemics. But as we know, time will tell. How will it go? Any opinions? Any thoughts?
On
State of the Union, CDC mask mandates, climate change, IPCC report. We've got a big bunch of science policy things going on.
I mean,
right
the
here.
mask thing, it depends what the goal is, I think. So I heard what's her name? The press secretary. The press secretary, Psaki. There we go.
Psaki, yeah.
But she talked about, I think actually both of them did talk about giving people a break, right? And so I think they both are being very clear now and saying, you're going to have to put the mask back on. We're just trying to give you a break, which I don't know. It feels odd to give up partially. We're going to give up and let you take your mask off, but we're still going to try to catch up again later when that causes a later surge.
Yeah, I had heard from many people online, epidemiologists, public health experts who are upset about the level at which it was deemed all right to remove the masks. But as Walensky said today in the White House press briefing related to the pandemic preparedness plan, it's different the numbers now are different in comparison to numbers, say, last June or July, because now we have more people vaccinated. Omicron is less severe. We have generally less severe. We also have more treatments like Pfizer's pill, the antibody, the antivirals, antibody neutralizing agents. We've got a lot better therapies, and people are not ending up in the hospital as much anymore. And so they are saying that their judgment for the level at which they deem it okay to remove masks is different now than it used to be. And I think that's the hard part, is because if you're trying to follow along with just the numbers, it looks like it's much worse than it was even 6-8 months ago still, yet the outcomes are different. And so it is a different situation. And I think that's part of the problem with the entire pandemic is there's not been a way to just look at it in a single layer. It's got multiple layers of complexity that all tie together.
Yeah, it's nuanced and
yes. And if you're not an expert in that area, you don't know why necessarily. Yeah. So what's happening here is very interesting, because we had the everybody work together, let's collaborate and try and protect everyone approach, and now it's the just protect yourself, whatever your risk is good approach. And this doesn't work well also if you put it in parallel or in comparison to like what's happening with climate change and the efforts we haven't done so far, which have been like, oh, do it yourself. And now we have to push towards, hey, everybody, we all really need to work on this together. Let's get the government involved. Let's get all this stuff, regulations, and let's make this work. So I don't know. There are a lot of parallels here, a lot of differences in the way these two scientific issues are playing out, though, that I find very interesting. And we'll see where it goes. But I wanted to jump from here to Blair's story.
Oh, yes.
He has a mask story.
Yes.
Next.
Yes. So speaking of these disposable face masks that everyone should be wearing, they're destroying the environment. So
it's bad.
Here's the thing. We've talked about the litter from face masks before on the show, but this is about the chemicals leaching from face masks in tide pools. Yes, this is
that's just the picture. The picture of a tide pool full of masks.
Tide pools.
So, first of all, important to remember that any trash on the ground can eventually make its way to a waterway, right? So any litter can end up in water. And once it ends up in a river, it can end up in, or through a storm drain or whatever, can end up making its way to the ocean and get caught in a tide pool. But also, there are also probably just people throwing them on the ground at the beach too. Let's be real. So, quote, we're seeing more and more masks in rocky pools, says Laurent Seron, a marine ecologist at the French Centre National de la Recherche Scientifique, who will present the new research coming up at a meeting tomorrow of Ocean Sciences. This could cascade up the food chain and up to us. They are seeing altered behaviors in tide pool animals that appear to be associated with the chemicals leaching from disposable masks. That includes signs of stress, reduced ability to detect mates, and reduced reproduction. And so all of those things are coming most likely from the plastic polymers, polypropylene, which is what masks are made out of. Yes, masks are made out of plastic. And so that same plastic polymer has previously been shown to have negative effects on aquatic organisms, so it's not surprising. But so they actually looked at 3 different types of animals: blue mussels, snails, copepods. They all had adverse effects to the leached chemicals. All of them were different. Blue mussels showed a stress response, moved away, gathered together, aggregated, looked like they were freaked out. Snails, they were less vigilant after being exposed, so they actually messed up their stress response, which increased their likelihood of being eaten, which also then brings the chemicals up the food chain. And then copepods had trouble reproducing because of it. They couldn't find the pheromone trails of females. So all that to say, those disposable masks are not just yucky litter, they're also causing chemical issues. So to Kiki's point, the thing that I was thinking about with your IPCC thing is that it really feels like society was getting really concerned about climate change, and then the pandemic happened and it was like, forget about that. We got to worry about this. And so we really have to figure out how to do both at the same time because they're connected. The environmental issues that are going to kill us and the pandemic-based issues that are going to kill us, they can work in tandem in bad ways. And I was actually just talking to Sodi about how all these COVID tests have all these plastic pieces in them, and it's creating litter, and it's creating more plastic waste. And I understand at a certain point you have to throw caution to the wind for human health, but after you have a second to assess how you're doing things, it might be good to
reassess
on the impact that you have by all these plastics that you're using in this pandemic. Because yes, most of these masks that are in tide pools are probably from inconsiderate people littering, but some of them could be blown off the back of a garbage truck or blows out of your dumpster after you put your trash very carefully in the dumpster. Or like I said, could be pushed downstream. There's a million ways that stuff could end up out there. Off of a garbage barge.
Garbage barges. We shouldn't even have these things.
You know we do.
But we do. Yeah, and along the lines of the face masks, there's a whole new modeling study that came out this week from Chalmers University of Technology that is called Modeling the Direct Virus Exposure Risk Associated with Respiratory Events. And it's based on trying to upgrade the old model that was developed for respiratory viruses back in 1934 and actually is too simplified to account for the complexity of transmission. And so this new model does that and takes masks into account. And it's another confirmation that masks actually do work. So even though the plastic waste is bad, masks are still good. And you can use them to protect others. So, so as long as we are in the pandemic, if you have a respiratory virus, you don't know what it is, maybe you have the flu, wear a mask. You can protect people. This is now, it's not just going to be a SARS-CoV-2 COVID-19 issue. It's going to be respiratory viruses kind of moving forward.
I sure hope they don't go away for that. I really wish people would wear them when they're sick forever after this. I don't want your cold.
I don't want your cold. I believe Justin has a story about microbes.
Yeah, this is Clarkson University researchers. They found a correlative link between energy levels and the gut microbiome. Associate professor of physical therapy, Ali Boulani, and his team searched the microbiome for associations between the microflora that make up the gut, the bacteria and such in there, and self-reported personality traits or mood ratings of 4 categories. They had mental energy, mental fatigue, physical energy, and physical fatigue. The 4 moods of 20 participants, which is not a big sample size, were assessed by survey, and the gut microbiome and the metabolome, which is the things that are being made by the microbiome, were determined from RNA analysis and some untargeted metabolics analysis, especially. So they found some of the most prominent phyla was only negatively correlated with physical fatigue. The second most predominant phyla that they found, Firmicutes, had members that correlated with all of the traits, which is an interesting hit. Bacteria Aniostrips was positively correlated with mental energy, negatively with mental fatigue and physical fatigue, respectively. So they're starting to now say, okay, hey, these people are saying they've got energy and that's correlating with a specific bacteria in their gut. Diet influenced the gut microbiota composition of the participants, and only one food group was correlated throughout all 4 of the different moods, seeming to affect all of them based on this one food group, which was processed meat.
And it was great, right? It totally was good for energy and all. Like it was that bacon and that salted ham.
Positively with mental fatigue and positively with physical fatigue, meaning it had the processed
it'll make you
meat,
take a nap.
It affected the microbiome, and that created mental fatigue or was correlated with mental fatigue and physical fatigue. And negatively, meaning if you didn't have it, you had a higher chance of having less of the bacteria that were leading towards or that were deficienting mental energy and physical energy. Anyway, it's just a correlative with a small sample size, and even the scientists are like, we need a larger study to confirm and explore this further. But they were a little further. They found the amino acids and enzymes and cofactors that are produced by the gut microbiota that were correlated to these different things. This is quoting Ali Boulani. These new findings support my previous work where we found that feelings of energy are associated with metabolic processes, while feelings of fatigue are associated with inflammatory processes. Since we're still learning about the gut microbiome, we don't know whether if we try to change our personality trait, we might see a change in the microbiome, or if we try to change our gut microbiome, would we be then able to see the difference in the personality trait?
That's what I was wondering too, yeah.
So, and it's there's a study that I was going to have last week that we didn't end up doing because I didn't end up doing the show, which is they had to do one of these studies on heart patients where they had looked at, hey, here's a gut microbiome configuration that we see that correlates with heart disease. And what the problem was, they ended up finding that half of that which was correlating with heart disease actually correlated with being on a heart medication. So previous to that, it wasn't. And then there's a lot of noise in these things. Is what I'm saying. Is that you have to suss out what is causing what's coming before, what's coming after, what's being influenced by diet versus mood. Is that influencing back and forth? But I found that to be an interesting first survey to maybe drill down and find those connections between our diet and energy level that aren't the food. They are the food. Mm-hmm. The nutritionist is right. The processed meat is what's making you sluggish, but it's not the meat's fault. It's what it's feeding in your gut.
Right. It's what populations it facilitates growth of.
So it is the food. Yeah. It's just the food is the first domino.
The mechanism. The mechanism has been missing. So then you might, then you could possibly have with this, you could have a probiotic enriched bacon. Right.
But you would have to keep up on it. Right?
Maybe you have to balance your bacon with probiotics. Maybe the problem is the people who are taking the bacon don't take the probiotics. To balance it out.
You gotta put in some leafy greens in there with your
I don't know. Just get yourself some Benefiber. You're good to go.
Is that what really this comes down to? Is eat a salad?
Eat a salad.
No, it does. And actually, a lot of the microbiome studies do come down to actually, if you eat a salad, it seems to fix everything.
Yeah.
That was a
2 cups of leafy greens at every meal.
That was a footnote to the heart study last week, which is that they found just positive correlation with the leafy greens and the lack of microbiota that led to heart disease. But then again, is it because the leafy greens do something, or is it because you ate so many leafy greens you didn't have a chance to eat the bacon wrapped bologna that was deep fried?
Did you wrap the bacon and bologna in lettuce leaves? Very keto. I don't know. All right, everybody. Moving away from the myths, let's move to bugs. I mean, it might not be processed meats, might not be your fiber intake that you're looking for, but researchers have, and we've talked about it on the show a few times, researchers have suggested that we should eat insects for our nutrition, right? Good idea. We should be eating the bugs. Bug biomass is better on the planet in terms of the use of nutrients and the release of carbon dioxide into the atmosphere. And it's better for us. It's high in protein, high in lots of nutrients, but that's not what this new study is about. This new study is about another reason why we should be eating bugs instead of the cows and the pigs and the chickens and all those things. Mm-hmm. It's for the plants. We should eat the bugs to help the plants. This study
so then there's less bugs eating the plants if we just ate all the bugs ourselves.
We're not eating wild bugs. We're eating farmed bugs.
These are farmed bugs, exactly. And as Blair has brought up, yes, these are farmed bugs. And these farmed bugs, instead of eating the bugs, we can eat the plants too. But it's the bug poop because you're farming the insects to eat. They produce a lot of waste. And they also have their exoskeletons that come off, all this frass.
I love that word.
Frass is the waste of these insects. The frass is highly nutrient rich as well, and it's full of nitrogen, which we apply to soils in the form of fertilizer. And maybe we wouldn't have to apply all the excess fertilizer and have all the use of ammonia and the creation of the fertilizers if we used the frass So if we farmed enough bugs, if we got enough people eating insects, there would be enough frass to then fertilize the plants, to then supplement the growth of the plants, which we could then eat ourselves and also use to feed more insects, which we could then eat. It is the cycle of our future diet, agriculture.
Hey, if you blend them up real small, I don't care. I just don't want to taste the legs.
Don't eat that. Yeah, the legs. Those little legs with little hairs.
Now we got to figure out what bug eating does to the microbiome. Because now I'm like wondering, the first step when you started talking about this, because I just been thinking microbiome.
I had the microbiome story. Mm-hmm.
I was like, I wonder if I ate nothing but termites for a year, if I could digest wood.
I bet your microbiome would be very similar to someone who eats a lot of shrimp.
Probably. The one issue with insects is people who have a shellfish allergy may be allergic to these insects because there are similar proteins in the chitinous exoskeletons of the insects, as well as shrimps, shellfish, crustaceans, and so on. So this is a path toward more sustainable agriculture if we only would eat the bugs.
Well, give cricket tortilla chips a try. I've had them. They're pretty good.
Look, I don't need almond flour. Just get me mealworm flour.
So that is also a thing. You can buy mealworm flour.
Terrible for the environment. Get me the mealworms. Come on.
Mealworm flour. It's gluten-free, I'd assume. Probably not. It's probably mostly normal flour with some mealworm in it. I don't know.
I don't think so.
We'll look it up.
Sure.
Yeah. We can talk about it in the after show.
We'll talk about this in the after show. Yes. Let's find out what's in mealworm flour. Stay tuned to the after show or watch the YouTube video if you want to know. All right. Cancer. It's cured, right, Justin?
Oh, hey, Stephen, I got the segment. Let's see. Just good news. The science news segment that takes on the worst possible topic and delivers just the good news about it. Cancer edition. Cancer has been cured in mice. Rice University bioengineers have successfully eradicated advanced stage ovarian and colorectal cancer in mice. And the treatment is fast. It took about 6 days. Removed tumors in 100% of the animals with ovarian cancer and 7 out of 8 of the animals with colorectal cancer. And this isn't one of those over the potential infinite horizon could open a door to a path for future researchers to... No, they're talking about human clinical trials that could be happening this year. And that's because the design only included components that had previously been proven safe for human use. And the method of attacking the cancer is already FDA approved. What they did, though, is they created a mechanism for the delivery. Researchers used implantable drug factory, is what they're calling it. There's beads about the size of a pinhead that were surgically inserted near the tumor that were then able to deliver continuous high doses of interleukin-2, a natural compound that activates white blood cells to fight cancer. Drug-producing beads were implemented with some minimally invasive surgery, and each contained cells that were engineered to produce the interleukin-2, which was then encased within this protective shell, so it did sort of a time release thing. So you put in enough of it, and every day this interleukin is being generated by these engineered cells, and it's right near where the tumor is. So in the newly published study, researchers placed the drug-producing beads besides the tumor within the peritoneum, which is a sac-like lining that supports intestines, ovaries, and other abdominal organs. Placement within that cavity concentrated the interleukin-2 within tumors and limited exposure elsewhere in the body. So that's the delivery system right now is like an IV. So this is quote voice of Amanda Nash, who's one of the authors of the study. If you gave the same concentration of the protein interleukin 2, through an IV pump, it would be extremely toxic. With the drug factories, the concentration we see elsewhere in the body away from the tumor site is actually lower than what patients have to tolerate with an IV treatment. Thus, the high concentration is only at the tumor site. Nash said the same general approach can be used that they used in this study, could be applied to treat cancers of the pancreas, liver, lungs, other organs. Drug factories could be placed next to tumors within the lining that surrounds those organs and most others, she says. And if a different cytokine is needed to target a specific form of cancer, the beads can be loaded with engineered cells that make that immunotherapeutic compound. They haven't invented a new drug. That's not dependent on new drug discovery. This is about proper delivery without attacking the whole body system, but actually placing something that can express the needed compounds directly into the surrounding area. Bead's outer shell is a cytokine-producing cell the bead's outer shell shields the cytokine-producing cells from the body's natural immune system. Cells are made of material the immune system recognizes as foreign objects, but not as immediate threats. So that's how they can surgically embed this in the body. It doesn't just reject and attack it immediately. Because it's like, that doesn't belong here, but it's not on the high threat list, so it doesn't get attacked immediately.
Right. It doesn't have like antigens that are being presented that it's like danger. I must fight it off. So
in case, there we go. Cancer, many forms perhaps, have been cured so far in mice, but I like the fact that this is they specifically endeavored to not break new ground on any of the compounds or the construction of their beads when they bioengineered this, so that they could avoid that whole situation of, well, that sounds really great, but now we got to study it for a decade to figure out if the thing that you're using causes a problem. They specifically created their design parameters around things that already are approved and okay, so that it could fast track. So
it's a mechanism of delivery that's very specific, that avoids the systemic effects, that keeps it very localized.
Yes,
so that so the drugs, the cytokines are super localized by the tumors, and the drugs can get in there.
Yeah,
the treatment can get right there.
And also a little bit of this location. What would you call it? The way that it's surgically located within the lining of specific tissues to prevent it from also escaping. So it's a little bit of a combination of those two things put together, and pretty amazing results. These were advanced stage cancer mice, so it wasn't like they gave themselves the hard challenge in the test and passed with flying. So,
yay!
Really
the mice passed the test. Now we have to see if it works in the pigs or the sheep, and then it'll go to, or maybe the primates and the humans.
We can go straight to humans with this. Everything on this list of compounds and the drugs and everything is already FDA approved.
Mm-hmm.
So now you just got to see if it works. Yeah?
It's just the mechanism. Yeah, see if the delivery system, the beads do it. Well, these little beads, they might not cause problems in the body environment, but like Blair was talking about earlier, we're throwing all sorts of things into our environment, like plastic masks and other things that have these effects that we don't really expect them to have. And researchers from Oregon State University, Suzanne Brander, who I've actually talked to previously on the show about her work into microplastics, she and her graduate and postgraduate students and postdoctoral scholars took a look at tire-produced microplastics. And so in the laboratory, they created micro-sized tire particles from tire tread to be the equivalent of tire tread that would be released on roads. tires lose massive amounts of their tread over the lifetime of a tire in these little tiny particles. We're used to seeing the big giant tire chunks that are left when a semi-truck blows its tire. But in reality, every time you drive, there's erosion taking place, and there's little
That
bits
is true. We're basically driving on like pencil erasers that are just slowly getting worn down.
Yeah, slowly wearing them down.
Why do we do that?
It's poor planning on our part, really. And this study goes a pretty long way to suggest that we really need to fix that, because they took this plastic, the microplastics, and they looked to see what these not plastics, but these tire tread plastics, rubbers, did in estuary ecosystem models and also freshwater ecosystem models. They have a couple of papers that are out. And in the estuary ecosystem paper, they found in their organisms, inland silverside and mysid shrimp, they found that both organisms, after being exposed to the tire tread particles, had significant, and this is at concentrations of these tire tread particles that are detected in the environment, had significantly altered swimming behaviors, such as increased freezing, changes in positioning, and total distance moved, which the researchers note could lead to an increased risk of predation and challenges for these shrimpy organisms to find food in the wild. They also had reduced growth depending on the level of the exposure to these microtire particles. And fish exposed to nanotire particles had reduced growth as well. They say leachates affected behavior but did not impact growth in either organism. The freshwater ecosystem paper, they looked at embryonic zebrafish and the crustacean Daphnia magna, model organisms in the laboratory, and they found in these freshwater organisms, they experienced mortality and developmental abnormalities due to tire particle and leachate exposures. Tire particle leachate was the main driver of toxicity for both organisms, and exposure to nanotire particles enhanced toxicity in comparison to leachate alone. So they found the toxicity from tire particles in all these organisms in these different ecosystem models, but sensitivity was different between the organisms in the different environments. And so there's, yet again, complexity and nuance, and some organisms are more sensitive than others. And so that kind of understanding could lead us to develop different risk assessments and policy decisions moving forward. And something that has been happening around my neighborhood over the last few years, the researchers mentioned several ways to limit tire particles from entering the environment, and one of them is installing rain gardens. I looked up what rain gardens are, and these are they're areas full of soil and plants that are available for in the landscape that collects rain from a roof, driveway, or a street and allows it to soak into the ground. They're often planted with grasses, flowering perennials, and they can be a cost-effective and beautiful way to reduce runoff from your property. And because it's soil, it helps to filter pollutants out in runoff situations. So there's also, if you're using perennials, it's also potentially pollinating insect habitat, songbird habitat, wildlife habitat. There's all sorts of pluses to these rain gardens.
Until somebody does a study saying what all the micropollutants are doing to the insects that are getting into the
they're getting all over the
birds,
yeah.
they end up into the.
Well, right now we are riding on the science, riding on 9. Route 66? No, I don't even know where I'm going with this anymore, other than to say thank you for being a part of TWIS today. Thank you for joining us. Thank you for sharing your time so that we can share the science with you. We hope that you are enjoying the show. If you are enjoying the show, please share us with a friend today. All right, we are going to come on right back. This is This Week in Science, and it's time for Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels and a motor.
What you got, player?
Oh, I have so many primates today. Do you want to hear about learning primates or sleepy primates first?
Primate.
Oh,
All the primates. And all of them at once.
Okay, all right. All of them at once. Sleepy learning primates. I'll tell you about my preferred story first. So this is about
why did you even
napping.
ask? Why did you ask?
It's
If you
because
were
I'm going to tell you both of them either way. You're going to have to listen to both of them no matter what. So, sleeping in nature. A lot of people look at animals in nature, especially animals that are higher up on the food chain. You say they sleep more than humans, right? They sleep a lot. They nap. Well, a long time ago, a study came out, a bunch of studies actually, that identified you can't catch up on sleep. You don't have a sleep bank, right? So, like, if you miss sleep for a bunch of days in a row, you can't then sleep all day and fix it. Similar, you can't sleep all day today and then miss a bunch of sleep for the next 3 days and be even. But one thing that does happen...
Does that mean if I miss a night's sleep, that I'm never going to recover from it?
It means you
Forever,
so, okay,
you're ruined.
Because at
So
some
this
point,
is-
it's fine. I got a problem with that. You can't recover
Let me
from
explain the difference. There is another theory called sleep homeostasis. And so in that, if you had a bad night's sleep last night, you're going to need a nap today. Okay, so like within the confines of a singular sleep cycle, a singular circadian rhythm, your body has to catch up to its expectation of sleep, or it should,
Okay.
right? And so the expectation is you see that in animals as well. It's not that if you miss a bunch of sleep a bunch of days in a row, you have really bad sleep a bunch of days in a row, you can then catch up on everything you missed. No, each day pretty much is kind of a clean slate.
I think we should call this second one the sleep singularity hypothesis.
Yes, sleep singularity hypothesis.
So then,
And-
hang on. So then
Yes.
that would mean then if I missed 2 nights of sleep,
Mm-hmm.
on the third night I could get 1 night of sleep and I'm all caught up.
Correct.
Yeah, that's a much better system.
Yes.
Then it's not
So-
that you can't catch up. It's
You
that
can't just pick the one you
absolutely
want.
can.
What's the
Right.
evidence,
It's
Claire?
that each day is a new day, basically.
Yeah.
So
Well, the
previous
same
studies
day,
of sleep
really, but yeah.
have revealed that animals of every species, from honeybees to humans, put aside a portion of their day to rest. A lot of animals nap. With some notable exceptions, all of these sleep studies were in a lab. So you're really missing part of the whole picture. And so this was, as far as these researchers know, the first study ever to examine sleeping behavior in a wild group of primates. So they were able to do this really intense sleep study on primates in the wild, interacting with each other to see if this sleep homeostasis is a real thing. So what they did is they collected high-resolution movement data from GPS trackers and accelerometers attached to the baboons, in almost every single baboon in this troop. And they found that baboons experience shorter, more fragmented sleep when sleeping near more of their group mates. It's like slumber party syndrome, right? Up all night. However,
Too exciting!
they also synchronize periods of nocturnal awakening with nearby individuals, suggesting that baboons might actually be interacting with each other and strengthening their social bonds. So they had these collars, the accelerometers on, they had GPS trackers, they used cameras, they did all sorts of things that were non-invasive to try to figure out what was going on with these animals. And so what they saw was that because these animals were highly gregarious, they were balancing their physiological needs for sleep with social pressures of group living. So they basically, what this is saying is that even after sleeping poorly, they would spend time on other priorities. Socialization, number one, for sure. Looking out for predators. These things are more important than catching up on lost sleep, than napping or sleeping during the night, right? And so this kind of calls into question a lot of previous sleep research on animals because it was kind of done in a vacuum. And a lot of previous sleep research also involved more invasive methods. And so if you had to use something where, for example, you needed to drill into their skull to look at their brain or anything like that, that is going to impact your ability to have them in a space with other individuals, which then is going to impact how they actually respond to these things. So basically what these studies are showing, sure, in a vacuum with no other stressors, if they got a bad night's sleep the night before, they will sleep more during the day. But if you put in all of the normal stuff that goes with being an animal in the wild, sleep is not number one on the list. And so this is looking at this idea that humans are kind of individual or unusual in the fact that we push sleep for other priorities. We have to go to work. We can't just nap in the middle of the day, or at least, I can. I don't know about other people who are watching or listening, but they're competing priorities.
The pandemic allowed a lot more people to nap in the middle of the day and do their work at different schedules.
For sure. But yes, there are these competing priorities that have us accumulate what you might call a sleep debt, although, for what it's worth, that may or may not exist, depending on what kind of debt you're talking about, and that this might be somehow unique to modern industrialized societies. But looking at what they're seeing here in these baboons, it looks like this actually has a much deeper evolutionary root of being able to kind of weigh costs of missing out versus sleeping. So if you sleep and you miss important social interactions, or you're not there to alert the group to a predator, or you miss out some grazing opportunity or whatever it is, the cost actually outweighs the benefit in a lot of cases. And so you'd be better off to be tired, but to be able to get all this other stuff. So you could also look at this like FOMO in the wild, I suppose.
That's what I was going to say. I mean, this sounds like these primates, or are we talking about teenagers or freshmen in college?
Yeah, absolutely.
What are you choosing? Are you going to sleep? Are you going to stay up and study with your friends? Are you going to go to that party? What are the more important things to do right now?
Yeah. Well, I would argue it would be the studying one, but the teenager might argue it is the party. You know what? Maybe they're right. Maybe I'm not right. Maybe the social interaction is more important. Who's to say? I mean, baboons would probably agree with the teenager in that case. The social interaction is the most important thing. But anyway,
did you just call all teenagers baboons? Because I kind of agree with you.
No,
I kind of agree with you.
No, I didn't.
They do operate very much like a baboon troop.
No.
Babeenager. I mean,
oh boy,
Tina Boone.
Oh no. Well, anyway, so yes, so this is something that the first study of its kind identified the need to do sleep studies in the wild, but also identified a new methodology that is less invasive. Accelerometry-based methods that are easy, they're cheap, they can be integrated into other studies that are used tracking animals in natural habitats, and then you can expand this sleep study across a huge range of species. And so this could also potentially be applied to humans. And so there's lots of different things that you could do to kind of figure out what's going on. But
I want to see them apply these collars to magpies.
Oh yeah, they'll pull them right off. I thought you were going to say
exactly.
Humans, and I was going to say most of us have them already.
Yeah, our smart watches, our smartphones, there are sleep trackers. You can stick little pads in your bed to track your sleep if you want. There's all sorts of devices these days. The one thing I really love about this, it's even though it's a small study, kind of proof of concept.
Yes.
It's interesting in that it kind of upends a lot of ideas about the importance of sleep, but at the same time, it also, I think, we so much recently, we talk a lot on the show about the importance of sleep. And if you don't get all the right sleep, your metabolism is going to be messed up, and that's a problem, and you're going to die young, and metabolic disease and all this kind of stuff. But maybe we can relax just a little bit.
Yeah, or maybe we can find a way to be more relaxed with our schedules a little bit when it's available. I think that's the other problem, right? Is that we are in this very strict day. The baboons don't have this strict day. They don't have to be at work at 9 a.m. no matter what.
Yep.
If things are chill, if everyone else is asleep, nothing interesting is going on, they can snooze. And that's the difference, right? Is that they can adapt to the need of their environment, but we have these very rigid, structured days that. So basically, the study is saying it's not unusual for animals to forego naps like we often have to do as adults.
Yeah.
But I would argue what is unusual is that we're not allowed to nap when it would be okay to nap. Like if you're having a slow day at work, there's no emails coming in, I finished my projects early, I'm going to take a nap. I mean, some tech companies allow that. I'm pretty sure they have nap rooms,
but we don't have a nap
but they
room.
also have 18-hour days.
Yeah, that's also true.
So
there's no TWIS nap room though, Blair.
No.
Next story. Come on.
Story.
Now.
So this story, I bring less for the results and more for the experiment design because it's very strange. So this is a study from Central European University and Central European University's Department of Cognitive Science, but also TWIS University, the University of St. Andrews, and Max Planck Institute for Evolutionary Anthropology. This is a very collaborative study. They wanted to know about how apes and other primates learn. Specifically, if they want to be taught. Because there are different ways to learn. So one way would be to observe someone doing something and then copy them. This is how we observe a lot of animals learning. Observation. This is faster than discovering it on your own. Their example in the article is, in case of a baby, randomly pushing buttons of the toy to see what happens, right? So just experimentation. But the best way to learn, most of us would argue, is when someone can teach you. They can say how to do something, why it works, and what doesn't work. And so the question is, do apes and other primates like to be taught? Human children show a preference to learn through what they call communication. That's not surprising. That's how our whole society is based, right? Is on teaching our children things. Primatologists have recorded several cases regarding the ability of great apes to learn from observing others. But what apes lack, or at least what current research implies apes lack, is intentional teaching. What they've seen is juveniles will learn from their mother how to use a wooden stick for a termite fishing situation just by watching her. But they say we have not seen mothers try to call attention to the juvenile to the most important parts of the actions. Like, look here, I'm going to do this way because XYZ. So do apes care about being taught or not? So right off the bat, I would say just because we haven't seen it doesn't mean they're not doing it. But I will shelve that for the remainder of this. So in this study, this is their attempt to try to identify whether primates had a preference to be communicated with while they learned versus just observing and copying something. So this is a ride, so stick with me. So they showed chimpanzees, bonobos, and orangutans how to operate a food dispenser device. In order to get the food, they had to insert an object into a small hole on top of the device. After they inserted it, it gave a sound and a food pellet was dispensed. That is, if they put the right object in. There were also wrong objects provided, which if that happened, then just nothing happened. They didn't get a sound, they didn't get a treat.
So it has to be right object, right port.
Mm-hmm.
Yeah. So you're learning multiple aspects of the situation.
Right. And so then they had researchers show the apes how to use it. They had two kinds of demonstrations. You can see where this is going. They had a teaching and a not teaching, right? So while one demonstrator was communicating with the ape, they would insert objects. So they would make eye contact, they'd clap their hands, they told the ape hello, they kind of mimed like they were teaching. The other demonstrator did not communicate. They only made some attention-grabbing sounds. They knocked on the floor just so the ape was looking in the right direction, but otherwise did not communicate, did not make eye contact, just went about their business. And so they had these very different procedures. Now, what I was trying to figure out is they didn't these two individuals doing this in different ways didn't do the same thing with the objects. The demonstrator who was communicating only used wrong objects. The demonstrator who was not communicating only used correct objects. Why would you change this variable? And I think this is why. Because if you did right objects with both of them or wrong objects with both of them, you would be unable to separate out the difference between the two, and they could just be copying. Otherwise, there's no way to separate out the variable of communication.
Yeah.
So they did these two different objectives so that they could see, and this is what they saw, that the apes who watched the communicator, the teacher, put wrong objects more. So they paid more attention to what their teacher was doing. Yes, absolutely.
Yeah. Oh, it didn't work. It didn't work. I don't want to do that.
Right. So actually they actually continued to put the wrong objects in. Even though they could see with their own eyes that the wrong objects never made food, they still chose those objects often. So
they did just copy.
They did, but they copied the communicator more.
Yeah.
So that is what's happening here, is that they paid more attention to the person who was talking to them, even though it didn't get them their desired result. In control groups, when either both demonstrators were communicating or none of them were communicating, but one was still using the wrong objects, the other one was still using the right objects, the majority of them did not have a problem choosing the right objects. They were able to do it and get the treats. So this is super confusing, but they actually did a pretty good job of experiment design to try to separate out these variables in a situation where we don't speak the same language. We don't know how they communicate fully with each other. We can't do that with them, but we could figure out a way to kind of parse out these different pieces of the experiment. So as opposed to the control, they copied the communicator, the teacher, more, which means they paid more attention to what they were doing. So they are sensitive to communication cues, and they can recognize them and react to them. So in the case of children, the assumption is that there's language involved, right? They're learning the language. They are cued into you. Look, this is how you should do it. It would be a tough kind of leap to go straight there with these apes after this one study, but it does look like, at the very least, they pay more mind to someone who is connected to them, who is making eye contact and talking to them. So the hope here is that this is a starting point. There will be further research to help understand what could be kind of the missing piece to make this true education, I guess, or teaching opportunities between humans and apes, but also between apes and apes. Because also, this is the other funny thing, is that in this study, they say that apes do not teach each other in the wild. And I have a really hard time believing that.
But because then the discovery is that it's not that they don't learn from each other. It's that apes could be better teachers.
They could be
better teachers than they are.
I mean, what they're doing is modeling behavior just by doing it. They're not sitting their baby apes down and saying, now you sit here and I'm going to tell you what you need to learn. Like, there's no deficit model.
When I was a lad.
And so they're not, and they do communicate with their child, their offspring, and with others, but it just happens along the way. It's not as so focused as we are as humans.
Yeah, you don't have to get them to put down the phone, sit still, and eyes forward to teach them something. It's always paying attention.
It's modeling behavior that works.
Education best practices now are not doing that. It's moving more towards the way the apes are learning. It's moving more towards learning by doing and experimentation and experiment first, theory later, as opposed to, how all of we were raised, which is sit
Read the
down,
book.
listen to this lecture, and then put your
Yeah.
hot dog in this solar cooker. The theory now would be send the kids out and do that, and then say, why did this work? Why didn't it work? And then learn together about all of the theories behind it, right?
Yeah.
And so I think to say apes, quote unquote, don't teach each other in the wild is silly. I'm so sorry. You did all this research with apes. I don't buy that. I liked the research that you did. They 100% teach each other in the wild. The way that they do it, maybe you're not defining that as education or teaching. I think it is. But it would be great to be able to observe different kind of copying behaviors in apes and say, okay, in this one, they made eye contact with each other. They groomed each other before or afterwards.
Yes.
In this other situation, they didn't do those things and they didn't carry on the information the same way, right? So we need to figure out how to analogize what this study is looking at into how apes actually communicate with each other.
Which we still don't know.
No.
So yeah, that if we
have
can't say
ideas,
that they don't
but-
teach each other if we
Yeah.
don't really understand their full modes of communication.
Yep.
Justin, tell me some science news.
You wanted science news. Oh gosh, let me see if I've got any of that left. Yes.
Yeah, you got it.
University of Copenhagen and Aarhus University looked at the spread of misinformation on the Twitter. University, so they were looking at COVID-19 pandemic face mask related Twitter activity between February to November of 2020. And they were focused on the acceptance or spread or rejection of misinformation on Twitter during the first big wave of the coronavirus pandemic in Denmark. Misinformation, they say, accounted for a small portion of the overall face mask related tweets. Very small percentage. With almost equal number of misinformation spreaders and rejectors. So in the early part of the pandemic, tiny little bit of people who are like, face masks make you inhale too much of your own exhale and that will cause brain damage. And then they had the same amount of people like, that's nonsense, right? By the end, though, the number of tweets rejecting misinformation exceeded the tweets spreading the misinformation. Over time, however, the tweets spreading misinformation outnumbered those rejecting it. So the misinformation grew during the pandemic. Most cases, tweets rejecting misinformation did not engage with substantive claims, but instead ridiculed the misinformation spreaders. Analysis suggests that future initiatives to limit online misinformation should consider also status-seeking dynamics amongst both the misinformation spreaders and the prominent misinformation rejectors. So they're saying the focus seems to be people speaking to their own audience and not actually engaged in a dialogue. 33% of all tweets rejecting misinformation used satire, irony, and humor, while that was only true of 8% of tweets spreading misinformation. So that kind of tracks. So that having a snarky sense of humor, I think tends to track more with people who are fact-based.
I think the other problem is that
experience.
is that we've been told for years now that conspiracy theorists don't care if you give them facts. But I think that's the problem is that you might not be swaying them, but it's about the other people reading it.
It's about
and it might be them.
It's not. I mean, it's the response, because I think what Justin said is exactly right. Is it's people playing to their own audience.
Mm-hmm.
You're trying to impress people who think like you. Oh, other people might be that think like me might see what I write, and I want to impress them. You're not actually. The people who are rejecting misinformation, rebutting misinformation, are not trying to have dialogue. They're not trying to actually have a conversation. And that's the problem. It's because
And
we're
that was,
all
and
putting ourselves on stage.
yeah. And it wasn't
Sorry,
all,
yeah.
but it was a big percentage of those
Big enough.
rejecting misinformation were basically just making fun of the people, which I kind of get. Like, I kind of get that. Like, that
I'll
is my
admit, it
first
makes
response
me
at this point.
it makes me feel a little better sometimes. I hope that doesn't make me a bad
Yeah.
person.
Yeah. No,
No,
it doesn't.
Blair,
It doesn't.
totally.
I'm not the one
but
making fun of them.
this is a problem.
But it's when someone else, posts a conspiracy theory, and I get to see the one little comment where I'm like, yeah, got it.
Yeah. So, but again, that's who they're doing it for. Is for you. Says Professor Rebecca Alder-Nissen, one of the researchers behind the study. We tend to believe that people eager to correct misinformation will be very fact-oriented. But our study shows that this group of people typically choose to ridicule those spreading misinformation. Instead of bridging gaps or inviting people to change their minds by updating their knowledge, their response to misinformation takes the form of know-it-all remarks intended to patronize their opponent and praise themselves. Consequently, the research has concluded a lot of people who either spread or reject misinformation are really trying to defend their own social position.
What I'm seeing from this study that's really important is that a lot of times on these social media platforms, we're not being thoughtful. We're trying to play. We're not talking. We're playing to our audience because what do we want? More followers.
Give facts, have a conversation with a conversational tone, or leave it.
Yeah.
I think that's the point, right? Is that if you are talking down at, reprimanding, patronizing these individuals, then that's not actually helping other people seeing that tweet, and you just look like a jerk. And then, and not to mention, that boosts the whole thing about Twitter, right? Is that boosts the tweet. Yeah. The more interactions it gets, the more people see it. So if you see a tweet that's nonsense and you don't want to spend the time to debunk it properly, maybe leave it.
Yes. Mm-hmm. Yep, absolutely. Yep. I'm super excited. The research that was done to determine the 12 individuals who are responsible for the majority of anti-vax information on Twitter, and the conference for science communicators that I'm involved in, Science Talk, which is happening at the end of March, the 24th, 25th of March. One of our keynote speakers on the second day is Imran Ahmed, and he is the CEO for the Center for Countering Digital Hate, which did that work. Oh. So I'm excited to hear him speak about that work. He's coming to Portland, so I'm hoping I get to meet him and talk about countering digital hate and misinformation. I'm very excited.
Very cool.
Yeah. Science Talk 22 for science communicators. Thank you. This is This Week in Science. We don't hate. We just enjoy science. We do. We like science. Mm-hmm. If you are enjoying the show, please head over to TWIS.org and click on the Patreon link and help support this show in an ongoing fashion. If you sponsor us at $10 or more per month, just per month, it's a fairly doable amount, we'll thank you by name at the end of the show. There's a lot more in rewards there as well. Thank you for your support of TWIS. We really can't do this without you. I'm going to come on back with a few more stories to finish out our Tight 90. Okay, let's jump into the brain. Mm-hmm. Blah, 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 blah. This study published this week in the Proceedings of the National Academy of Sciences is very exciting to me. It's researchers out of Carle, Illinois College of Medicine at the University of Illinois. Researchers worked on this multidisciplinary project to develop a way to visualize epigenetic markers in the DNA of your brain. So you know how an MRI, usually responds to the changes in oxygenation or the vibration of molecules, atoms within your brain, the spins of protons. And you can get the researchers can get a picture of activation in the brain, of blood flow in the brain, of things that are happening energetically in the brain. Well, they used previous research to that showed that a marker known as carbon-13 can be imaged in the brain. And carbon-13 could be used if it was given in the diet to a person. In this case, it was pigs, but could be given in the diet as a way to mark methyl groups, which involve carbon, these methyl groups that are needed for DNA methylation. So what happens when you experience something? There are a lot of changes that take place within the molecules that are inside your brain. Not only do your neurons send their action potentials all over the place to signal with each other, the neurons themselves are also computing what's happening, and there are changes to the DNA that will change the way that the DNA responds the next time you have a similar experience. It's part of the learning process. And the DNA methylation is it's a methyl group, this molecular group that it gets attached to the DNA to kind of wrap it up. And where the wrapping happens, it changes the way that translation and transcription of the DNA can occur. Translation and transcription is necessary for the production of proteins, which allow processes to take place in the neurons that allow behaviors to occur. So they used baby pigs in the first few weeks of their lives. They were fed with carbon-13 labeled methionine. And then as the pigs grew, without having to sacrifice the animals, they could put them in these, what they're calling eMRI machines for epigenetic MRI. They're able to image these epigenetic changes as the pigs developed, experienced, learned, and grew. Cool thing. I'm so excited about this technology. Being able to actually image molecular changes within the brain.
That is wild. But you have to be on the carbon-13 diet from a young piglety age for this to work.
Right. But I mean, it would be the kind of thing where if we wanted to do human studies with carbon-13, methionine, this is something we could have in our diets. It's not, it's an amino acid. It's an essential amino acid. So it's not a problem. And instead of using carbon-12, which is very common, you use a form of methionine that has this isotope that's a little bit different, carbon-13, that can be read a little bit differently. And so we could potentially use this technology if as they develop it further to see how the brain changes in response to certain experiences. So you could imagine like psychological experiments where different groups have different experiences. Watch a movie that's shocking or watch a movie that's calming and see if there are changes after having a particular experience. Yeah, you could imagine the work that you could do there.
Very cool.
On pigs.
On pigs, so far.
Or apes. Yeah.
I mean, it's like almost humans. Aren't we just long pigs?
Yeah.
Pretty much. Anyway, new technology. Yeah, new technology, imaging epigenetic changes to DNA in the brain. Pictures, changes to DNA. It's just...
That's wild.
Yeah. It's kind of blowing my mind a little bit. And then from the NIH, this came in very, very last minute that I was very thrilled about. Researchers at the National Eye Institute have been looking at organelles within the eye's photoreceptors. These little organelles you might recognize the name of mitochondria.
Oh, that's the energy center there, where it's all you got in the genes there. The
powerhouse of the cell.
Powerhouse of the cell, energy
Chromosomes are
center.
what happened.
Yeah, mitochondria. They're good. All cells have them, or they should. Very good for our cells. Anyway, this study found an entirely new purpose for mitochondria in the eye's photoreceptors. These organelles, they're not just there for energy production for these photoreceptors. They also are stacked in a way that has an optical effect. So what they've discovered using just a microscope, modified microscope, they observed the optical properties of living cone cell mitochondria that were exposed to light, and they found that the mitochondria focused, concentrated the transmission of the light from the inner to the outer segments of the cone photoreceptors, and thus act as focusing elements within the photoreceptors of the eye.
Wow.
So they used to just be like, oh, why do they have, the eye photoreceptors must use a lot of energy.
Mm-hmm.
Why are they stacked in that way? What's going on? Oh, look, they actually have a function in focus.
You had one job. Oh, no, actually, you've got more than
Mhm.
one. Oh, wow.
Mitochondria leave some jobs for the rest of them.
Nah, I'm going to do them all. I
It's-
could be mitochondria. Mitochondria is.
So if multicellular organisms didn't indeed come from single-celled organisms getting eaten by each other, then I'm surprised. I'm surprised that we haven't found more organelles that do a bunch of different kind of disparate things, because
I think we also just kind of go, oh, we found your function.
yeah. Oh yeah, for sure.
And it's like, that's great. Thanks. Good
Here's
job.
here's the label. Yeah, you're in the box. You have your own box there.
We love our boxes.
Oh, what if I got something else? No, I've already labeled that box. I got to move on. A lot of boxes.
Yeah, but if organelles really did come from engulfed single-celled organisms, it sure would make sense for more of them to do multiple things. I understand that over time it makes sense to streamline the work and do all this kind of stuff, but for it to all kind of go away and have it just have a very one clear rule just seems like it wouldn't always happen.
Yeah, one thing that I thought was very interesting, just from this, they in this story, just based on this research, they realizing that the mitochondria have this function, they compared it to other animals, and it does give, like you're talking about, like, okay, what are the other functions of different organelles and things? It has been discovered also that there are photoreceptors of birds and reptiles that contain oil droplets. And the oil droplets in the inner segments of the eye serve an optical role that might be similar to this mitochondrial microlens function. Yeah, so perhaps there were these oil droplets, and then the oil droplets got replaced. I don't know. There's a lot of ideas. The researchers say this insight conceptually bridges compound eyes in arthropods with the camera eyes of vertebrates, two independently evolved image-forming systems, demonstrating the power of convergent evolution.
Mm-hmm.
Power of convergence and science, and mitochondria are so cool. Oh, and by the way, I just want to finish this up with two quick stories of science working the way that it's supposed to work. No more brains. We're going to space, everybody. This week, Friday, there's a rocket that's supposed to impact the moon. I reported previously that it was SpaceX. Well, researchers looking at the trajectories of various bits of space junk from different launches think that it's not a SpaceX booster from a Falcon 9 that's going to hit the moon, the far side of the moon. It's going to be a Chinese booster that is from the Chang'e 5 T1 mission in 2014. Although the Chinese deny this is theirs. The rocket is going to hit what?
Oh, it's just like we got too much space junk up there. We can't even figure out.
So
Yeah,
Whose
this rocket's
it
gonna
is?
hit the moon. Oh, you put that one up there? I don't know. Is that one of mine? I can't tell. I
I'm
can't
not
keep track of all
cleaning
my
it up.
space
It's
junk
not
anymore.
mine, Mom.
Anyway,
And why
it's
are
going
they denying it? Why would they deny it? Like,
it
they're
does,
gonna
what
get
good
some
does that
moon
do?
damage liability issues coming up in the future. Oh, you hit the moon. You owe us. Who do you pay? Who do you charge? What would be who cares?
I don't know. The
I
rocket's
don't know.
going to impact into Hertzsprung crater on the far side of the moon at 7:25 a.m. approximately Eastern Standard Time on Friday, March 4th. We won't be able to see it because it's on the far side of the moon, but we know the trajectory, but we don't really know whether it's like spinning or whether it's going end over end. We don't know exactly what's happening with how it's spinning toward the moon, so we don't know how it's going to impact, but we have a good idea. And then we're going to send our lunar orbiter around to take a look at it and finish up observations and look at the atmosphere of the moon to see what shot into space surrounding the moon once the impact happened.
How long will it take the little lunar buddy to get over there? Do you think? Do you think we'll find out the same day?
I don't think that we're going to find out the same day.
Okay.
No. I don't think that's going to happen. Yeah, I'm not sure exactly when the lunar orbiter is going to be there to take a look.
Okay.
Yeah. I'm not the sooner the better, but I'm not sure.
Yes.
They're going to do what they can. And then in other, oh, hey, science is doing what it's supposed to kind of news. There was a report in 2020 from the European Southern Observatory that there's this black hole that's 1,000 light years away from us. It's the closest black hole to us. And wow, this is amazing. It's super close. HR 6819 solar system. And there were some papers, but then there was some contestation. They contested researchers.
Contestation?
I just made up that word.
I like that word.
This is some Belgian researchers said, hold on a second, what's going on? And so they came up with some new hypotheses as to what was going on. They got some new data from the very big and the very large Terra telescope. And they were able to, with this data, have finer resolution to be able to determine that it is not actually a black hole. What it is, in fact, is a vampire.
What?
A vampire binary system. It's two stars orbiting each other. One star has ripped all of the star stuff away from the other star, and they're just spiraling. They're in a death spiral. But it's not a black hole. So, no black hole 1,000 light years away from us. Thank you, science. We needed to know this. There might be others. We just haven't found them yet. But this is the way science works. Someone puts out a result. Someone says, I don't think so. Someone else says, okay, let's look at more data. And then the experiments are done and you get to a more accurate answer. And they collaborated. They didn't fight each other for these papers. The two teams came together and published this final paper together. So this is science at work. Thank you, science, for bringing us the knowledge. Did we bring the knowledge? Did we do it?
Yeah.
I think we did. Blair's been yawning for a while, so I think we...
Stop it. Why you gotta tell the podcast listeners about it?
The 90 minutes.
I've been yawning since I woke up this morning, so okay.
Yeah, why are you gonna put me on blast, Justin? It's so rude.
Maybe I had too much processed meat. That's not what happened. We'll see. Thank y'all for listening. Thank you for being here for another wonderful episode of This Week in Science. We really do enjoy having you here. It's time for some shout outs. Thank you too, Bada. Thank you so much for all your help on show notes and show descriptions and on social media. Our chat rooms. Thank you, chatters, for being here during the show and chatting and letting us know what you think all the way through. Identity 4, thank you for recording the show. Gord and Lore and others who keep the chat rooms safe places. Thank you for keeping them safe places. And Rachel, thank you for your assistance and for your editing. And now, publishing. Getting closer to publishing. More duties. We love it. And finally, thank you. Thank you, as always, to our Patreon sponsors. Thank you to Richard Badge, Kent Northcote, Pierre Velazar, Ralphie Figueroa, John Rataswamy, Carl Kornfeld, Karen Tozzi, Woody MS, Chris Wozniak, Dave Bunn, Vigard, Chefstad, Hal Snyder, Donathan Styles, a.k.a. Don Stylo, John Lee, Ali Coffin, Matty Perrin, Gorov Sharma, Reagan, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis 104, Skyleague, Paul Ronovich, Kevin Bearden, Noodles Jack, Brian Carrington, Matt Bass, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenfo, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard Brenton Minish, Johnny Gridley, Rachmi Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Bob Coburn, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim DePoe, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul D, Disney, David Summerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, and Jason Roberts. Thank you. Thank you all for your support of TWIS. On Patreon, please head over to twis.org and click the Patreon link on next week's show.
We will be back Wednesday, 8 p.m. Pacific time, or Thursday,
Thereabouts.
5 a.m. Central European time. Broadcasting live from our YouTube and Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast? Maybe while you bake some mealworm cookies? Just search for This Week in Science where podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links and stories will be available on our website, www.twist.org, and you can also sign up for a newsletter.
You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will end up in my mealworm cookie batter. It will get baked into cookies. I will eat it, and we will never read it.
Yum, yum, yum.
Yeah, you can also be a part of the audience as we ridicule unscientific people on Twitter, where we are at Twist Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes due in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science.